Very good evening, everyone. Please confirm if the screen is visible. Good evening, Sunshine. Good evening, Anusha. Good evening, Lakshmi, Arzu, Soumya. Good evening, Das. I hope you all are doing great. So, how's the revision going on? Okay, thank you for confirming. So, I'm Dr. Tanushree, and I welcome you all to another session of CSIR NET Power Hours Last Minute Revision Series. So far, already you have revised and practiced questions from Unit 1, Unit 3, Unit 10. So I hope it was really helpful to you all. Yes? Was it helpful? Please write in the chat. Yeah, very good evening, Sapna, Shivani. I can see some more have joined. Good evening, Hemlata, Krishna. Okay, good to know. That's great. So today we will be revising the concepts from Unit 8 and 13, as well as we'll be practicing questions from Unit 8 and 13. So the exam is approaching and it's high time that you focus on the revision part as well as practice as many questions as you can. So to help you all, to help you prepare better and to help you in assessing that how is your preparation, Biotechnica has come up with this CSIR NET Power Hours last minute revision series. So without wasting much time, let's start with the session. And for uninterrupted streaming of the session, I'll be keeping my video off. So see you at the end. I'll switch on my video later. So let's begin with the revision of Unit 8 first. So Unit 8 is inheritance biology, and I will be particularly focusing on the questions from the topic probability and genetic mapping. So first of all, we'll be revising the concepts from these two topics from Unit 8, and also we'll be practicing the questions. So let's get started. Now, why these two topics specifically? Because these are very important from exam point of view, and you can expect the questions from the probability in genetics, from the gene mapping, from the linkage. So here is a question on your screen, which is based on probability. So just go through this question, and then we'll revise the concept behind this. We'll go through the concept on which this question is based, and then we'll solve the question. Yeah, hello Saptala. Good evening, Shraddha. Good evening, Ria, Lucky. So are you all excited for Unit 8 and 13 revision? And please note that Unit 8 other topics like Mendelian genetics, pedigree analysis, and mutation, the revision of these topics will be done in another session later. Okay, right now in this particular session, we'll be revising the concepts of probability and genetic mapping. So basically, if you go through this question, the question is about the gene A, B, and C. They are independently assorting and controlling production of a black pigment, basically, right? So here, these are the three genes which are majorly being responsible for the black pigment. The pathway is also shown, right? So from colorless to black. So colorless conversion to black is being done by these three genes. Now, alternate alleles are also there, which are represented with lowercase letter. So A, B, and C, these are the normal alleles, and the alternate alleles, you can say, which are responsible for abnormal functioning, or you can say these are the mutant alleles, which are designated by lowercase letter A, B, and C. So the question is, when the black is crossed with the colorless, the F1 generation is all black. So you know this kind of cross, right? Mendelian genetics, we have studied that when triple homozygous dominant cross with triple homozygous recessive, so this is the parent generation, you get the F1 generation, which is triple heterozygous, right? So this is your F1 generation. Now, when the F1 is self, so that means you have to perform this cross, and you have to find out how much proportion of F2 is colorless. Now, the one way is you draw a Punnett square, right? You write down, you determine, like it's a trihybrid cross. If you see, this is a trihybrid cross. And in trihybrid cross, there are 64 offsprings which are formed, right? Now, one way is that you determine the gametes and then you draw a Punnett square or Folkline method you use and determine all the 64 combinations. But this process is very, very time consuming. So I will be giving you a trick. I will be giving you a formula, right? And before that, we'll first understand what is probability. So let's have a quick revision of what is probability. So probability means chance of a particular event to occur. And especially when it comes to genetics, probability rules are very, very important. So basically, in genetics, we use the probability rules to determine the chance of gene flow, the chance of passing the genes from parent to offspring. And also, we use it to determine the chance of gaining characters. That is, what is the chance of getting a desired trait in a progeny. So, probability can be calculated as the number of favorable cases divided by total number of possible cases. This is a very general formula. Like, for example, when we talk about childbirth, so you know that there are two possible cases. A child can be a boy or it can be a girl, right? But if I specifically ask you, so these are two possible cases. But if I specifically ask you, what is the probability of getting a baby boy, then you know it is 1/2. So, or if I ask you, what is the probability of getting a girl, baby girl? So it's a favorable case. So it is 1 and total possible cases are 2. So half, 50-50. And even we know, like the father has X and Y chromosome, mother has X chromosome. So mother can only contribute X, but father can contribute X or Y. So if both the parents are contributing X chromosome, then the child will be a daughter. But if mother is contributing X and father is contributing Y, then the child will be a son. So you can remember always that the probability of always getting a boy or a girl will be 50-50 only or half. Then let's take another example. Let's say you are crossing yellow seeded plant with green seeded plant. Now yellow seed is a dominant trait. So you know that the F1 generation, it's a monohybrid cross, right? So when you cross them, the F1 generation will be heterozygous and the progeny will be all showing the dominant trait, that is yellow seed. But when you perform a selfing of F1 generation, so this is a monohybrid cross, right? So you will get 3 out of 4 yellow seeded plant. So we are talking about phenotype. So when homozygous dominant condition is there or heterozygous condition is there, we'll get the yellow seeded plant. And 1 out of 4 will be the green seeded plant. So always the probability of a dominant trait, when you're talking about a monohybrid cross, the probability of a dominant trait or dominant phenotype is 3 out of 4. And for recessive, it is 1 out of 4. And I told you that in the beginning, I will be telling you a formula. So this is what you can remember. It depends upon the question which is being given in the exam. It's not that always you have to use this formula, but for the quick calculation, this will be definitely helpful. So always remember, if the event has a probability of P, all the other alternative combined will have a probability of Q, which is given by 1 minus P. So all these points you have to remember. Then let's quickly revise the probability rules, because I'm sure you have come across the terms product rule and sum rule. So basically what these rules are, and it is more important, it is to know when to use these rules. So first is product rule. It is also called as AND rule and multiplication rule. Dear participants, dear students, for your quick revision, I have specifically made this comparative table. So this will help you quickly revise the product rule and the sum rule. So what is sum rule? It is the addition rule, also known as either or rule. Now, product rule states that probability of two independent events occurring simultaneously is the product of each of their individual probabilities. However, sum rule states that the probability of occurrence of any of several mutually exclusive events is the sum of probability of the individual events. So I have used two terms here, independent event and mutually exclusive. So what is the meaning of independent event and mutually exclusive? So let's understand this with the help of an example. If I ask you, like I'm sure you all know about dice, right? Dice has 6 faces. So if I ask you, what is the probability of getting number 4 on 2 sequential rows? So on dice, you know, on every face, some number is written. So when you roll a dice, the probability of getting number 4 will be what? That is what we have to find out here. So when you roll a dice, either you can get number 1 or number 2, 3, 4, 5, 6. So possible cases are 6. But favorable case I'm looking for is number 4. So obviously the probability of getting number 4 will be 1 by 6 only. Now when you roll the dice again, I'm talking about sequential rolls, 2 sequential rolls. When I roll the dice again, I can again get number 4. So the probability of getting 4 again is 1 by 6 also, right? Again 1 by 6. Now according to product rule, the independent event, now these two are independent event. Why these are independent event? Because if you have got 1 by 6 in when you roll the dice once, and when you throw it again, when you roll the dice again, you can again get 1 by 6 because they are independent events. Independent means that it doesn't mean that if you have got 4 on one throw, you cannot get it again. When you play Ludo, it happens, right? You play a chance, you get number 1, then again you throw the dice, you can again get number 1, right? Similarly, when you're talking about the probability of getting 4 on 2 sequential roll, so because these are independent events, the probability of getting 4 is 1 by 6. And as we are talking about 2 sequential roll, so the independent events, we can now multiply to get the probability of the 2 sequential roll. Because according to product rule, it states that probability of 2 independent events occurring simultaneously is product of each of their individual probabilities. So you multiply individual probabilities and you get 1 by 36. So the keyword here is independent. Whenever in any question is related to independent event, you always have to use the product rule. However, what is mutually exclusive? If I ask you, or when I say that when you roll a dice, what is the probability of getting number 3 or 4? So this is either or condition, right? This is either or condition. Here it was and condition, right? Either or means that when you roll a dice, obviously the dice will either show number 3 or 4. It cannot show both the faces together in a single throw, right? So these are mutually exclusive events. So what is the probability of getting number 3? 1 by 6. What is the probability of getting number 4? 1 by 6. And because these are mutually exclusive event, you will add them. So when you take a LCM, you get 2 by 6. And finally, you will get 1 by 3. So just remember when to use product rule and when to use sum rule. Priya, you are absolutely right. And Sakina, thank you very much. Even I'm glad uh, to come today, and together we all are revising Unit 8 and 13 together. So even glad to see you all here attending the session. So coming back to the question, now I would like you all to go through the question. I've already explained you the question, solve it and tell me what is the answer. So you need not to take more than a minute, okay? Try solving it quickly. So others can solve. Meanwhile, Sandeep, you have asked me to explain sum rule again. See, Sandeep, sum rule is used in case of mutually exclusive event. Okay. I told you that when you roll a dice, what is the probability of getting number 3 or 4? So I told you when you throw a dice, either you can get number 3 or either you can get number 4. You cannot get both the 3 or 4 number altogether. It is not possible, right? When you throw a dice, it will show only one face, either with written number 3 or with number 4. So in that case, when it's a mutually exclusive event, you have to use sum. Okay, coming back to my question again. So I'm getting mixed answers. Some are saying option 3, some are saying option 2. So I gave you a choice that either you can draw the Folkline method or Punnett square and then calculate, because you have to perform selfing of F1 generation, which is this. So see, the trick here is always consider one gene pair at a time. Okay. So if I say one gene pair at a time, so don't you think this is a monohybrid cross? This is monohybrid cross and this is monohybrid cross, right? So you can write down the gamete of one parent on one side, gamete of another parent on another side. So homozygous A, heterozygous condition, and homozygous recessive A. Similarly here also, so you can quickly draw it in your copies. I'm sure all of you are keeping a notebook because at the end you have to solve the questions, right? Now you have to find out F2, which is colorless. So what is the probability of colored? 3 by 4? Because in homozygous condition, see, colored will be when the A allele will be in homozygous or heterozygous condition, that gives you color. Similarly, same goes with B and same goes with C, right? So colorless will be only when you have this condition, right? So I already told you that when you are performing this kind of cross, right? So first of all, you have to, according to the pathway, the color production, the dominant alleles of all three genes have to be present together, right? So that means genotype should be either, like you should have A homozygous or heterozygous, similarly for B homozygous or heterozygous, and similarly for C homozygous or heterozygous, right? Similarly, different combinations can be there. So probability of getting the black color is 3 by 4, right, for A allele. Similarly for B also 3 by 4 and for C also 3 by 4, right? So when you add, now because these are independent events, we use product rule here. That gives you 27 by 64. And even in the beginning, if you remember, I told you this formula, right? So 1 minus P, probability for black, give you the alternative phenotype. That is 1 minus 27 by 64. When you solve it, we'll get 37 by 64. So answer here is option 2. So those who have answered option 2, it is correct. Clear to everyone? So specifically, I told you this formula so that in the exam, if such type of questions are asked, you can use them. Was it helpful? Yes, can I get some yes or no in the chat? Was it helpful, students? Okay, that's great. Okay, that's great, Krishna, Shivani, Revati, Red, Coco, Sakina, Shweta, Lakshmi, Krishna. Thank you so much for your response, Rumesa, Soumya. Now moving over to another question. Now this is also a question based on probability. So solve it and tell me what is the answer here. The question is, a recessive inherited disease is expressed only in individuals of blood group O and not expressed in blood groups A, B or AB. Alleles controlling the disease and blood groups are independently inherited. A normal woman with a blood group A and her normal husband with blood group B already had one child with the disease. The woman is pregnant for second time. What is the probability that second child will also have the disease? Hemlata, when to use this formula? So see Hemlata, I told you that in this particular, the previous question which I told you, we were looking for colorless, right? So out of 64, how many will be colorless? Like you need to have all the three dominant alleles for the color to be there, right? Black color. So whenever any alternative phenotype is asked, in that case, you can use this formula. But always remember, every probability question is not same. Like here, that is why, in order to avoid the confusion, I'm giving you this another question based on probability, so that you can clearly get an idea that when to use which rule, how to solve different probability questions. Okay, please remember students, in the exam, like all the probability questions cannot be solved on the same pattern. Okay, you need to practice a lot to solve the probability questions, because every question can be different. Okay, so here I'm getting the answers. Soumya, what is your doubt? You can tell me. Soumya, you are right. You have mentioned that colorless chance is 1 by 4. But don't you think we are talking about the three alleles? For a single, it is 1 by 4. You are right. Okay, coming back to the question. So it is 1 by 2, 1 by 4, 1 by 16, and 1 by 64. So some of you are answering option 3. So majority says option 2. And some have answered option 3 also. Kavya, Sakina, you are saying 1 by 16. Shivani, okay, 1 by 16. Sandhya, Lakshmi, Shivani, Arzu, Shraddha, Priya, you all are saying 1 by 4. And even Coco, you are saying 1 by 2. Now see, if you read the question carefully, see the very first thing always when a probability question is there, just read it carefully. If you are not able to understand it, read it again. Okay, if you are not able to understand in reading once, read it again. Now think of or carefully note what all information is given to you. So it is mentioned that there is a recessive inherited disease. That means a disease which is associated with a recessive allele. And you know that when a disease is associated with a recessive allele, the disease can only occur when the recessive allele in homozygous condition, right? But the condition given here is that disease is expressed only in the individuals having blood group O. So two things are there. One is disease, another is blood group. And blood group O, so you know an individual will have a blood group O only when it has a allele lacking A or B antigen, right? Like we have three alleles for blood group A, B, and O. A and B are dominant over O, but that is A is dominant over O, B is dominant over O, but A and B display codominance, right? So if the individual is having A blood group or B blood group or AB blood group, the individual will not show the disease. It will only show the disease when it has a O blood group. So O blood group only occurs when an individual inherits both the O allele from the parents. Now it is mentioned that the woman has a blood group A and husband with blood group B. So that means we have to consider two cases here, one for the blood group and another for the disease. And when the probability question is given, you have to also see which rule you can apply here. So I gave you two keywords, either or, mutually exclusive, right? Or independent assortment, independent event. So here you can see a keyword is given independent. So that means we have to use a product rule here. So in solving this question, you have to consider two cases. One is probability of child having O blood group and probability of child getting a disease. So you know that when the mother, like it is already mentioned that mother is having A blood group. So why AO I have written? Why not AA allele I have written? Because a individual can have A blood group in these two cases. So it cannot be this genotype. Mother cannot have this genotype because if we are talking about the inheritance of the O allele, then and the blood group O in the progeny, that means it is only possible when male and female both are contributing O allele. And this is the reason I have considered this blood group over here. So in the progeny, either the blood group can be AB or B blood group or A blood group or O blood group. So what is the probability of getting O blood group? 1/4. Then what is the probability of getting a child a disease? Now again, as it is mentioned that disease is associated with recessive allele, so that means in the progeny, if the recessive allele is coming in homozygous condition, then only the disease will occur. So again here we have to consider heterozygous condition. Please note students that if in any question they don't mention the genotype of the parents, by default you have to consider them to be heterozygous. So again, what is the probability of getting the homozygous allele, the recessive allele in homozygous condition? 1/4. Yes. So probability for O blood group 1/4, probability of child getting disease 1/4. And these are independent events. Therefore, we will multiply these independent events. We will use the product rule here and you will get the answer 1/16. So here answer is option 3. So those who have answered option 3, it's very good. And those who have answered option 2, again, very good, because this will, like if you, unless and until you don't make a mistake, how will you learn, right? So I really appreciate you all for trying and solving the question. Very good. So I'm sure now those who have answered option 2 don't get disheartened, okay? Because this is how we learn. So I'm sure it is clear to you all how to solve the question. So now you get an idea how the probability questions are so unpredictable, right? It's not that every time you have to use the same strategy. Different strategies have to be used. Okay, now moving on to the question on genetic mapping. So please go through this question. Just go through it. We'll revise the concept of genetic mapping first, and then we'll solve this question. Okay. Escherichia coli cells were simultaneously infected with two strains of phage lambda. One strain of lambda had a mutant host range, is temperature sensitive and known to produce clear plaques. Another strain of lambda carrier, the wild type alleles, is shown with this. Always remember, participants, that dominant and recessive, like dominant allele is written with uppercase letter and recessive allele in lowercase letter. But in genetics, we have another way of representing the allele is like when it is given mutant and wild type. So instead of writing the uppercase or lowercase, you write the lowercase only. But if you attach a plus sign, that means it is representing wild type allele. And mutant alleles are again written in lowercase letter form only, but without the plus sign. So either minus or without the plus sign. Okay, without the plus sign, it is usually written. So wild type is usually dominant because it is most common in nature. Okay, so wild type. Now progeny phases were collected from lice cells and were plated on bacteria, and the following progenies are obtained. So I'm sure you know what are bacteriophages. They are bacteria eating viruses, right? And we have M13 bacteriophage, lambda bacteriophage, right? So here lambda bacteriophage is given. Now, in this question, you have to determine order of 3 genes and map distance between them. And here the options are given to you. So let me first tell you what is genetic mapping or what is genetic map. Okay, let's revise the concept first. So genetic map, like why do you need a map? I'm sure you have heard of geographical map, historical map, right? Geographical map, Google map, I'm sure you all use, right? To determine the location, right? To find a particular location, you use Google Map, right? If you have to go from your house to a friend house or to any grocery store, right, or any other place, you usually use Google Map to find, if unless and until you are familiar with the location. If you are not familiar, you use the Google Map. You even use it to find out which street you should take, where is the less traffic, where is the more traffic, right? Similarly, genetic map is used to determine the location of the genes on a chromosome. Now, there are two type of maps basically when you talk about map, genetic and physical map. So, genetic maps are based on recombination frequencies and they are measured in terms of centimorgan or map unit. So, these are the units for that we use for genetic map. And one map unit is always equal to 1% recombination. However, physical map, they are based on, like, they are measured in terms of base pairs, right? Now, basically, what is the meaning of recombination frequency? What is recombination? Recombination, where new combinations are produced, where new combinations you see, right? And the basis of recombination is crossing over, which occur in the during the meiosis 1, right? Now, there is a formula to calculate the recombination frequency, and that is number of recombination progeny by total number of progeny by 100%. So basically, the crossing over occur between the non-sister chromatids of homologous chromosomes, and it occurs during the meiosis 1, prophase of meiosis 1. And during the whichever strands are participating in the crossing over, then leads to the recombinant gametes. And the formula to calculate the recombinant frequency is mentioned over here. So, like for example, if I mention that gene A to gene B distance is 5 mu, gene B to gene C is 10 mu, and gene A to C is 15 mu, don't you think you can plot a genetic map? Right? Because A to B is 5 mu, B to C is 10, and 5 plus 10 is 15. So that means B gene is present in the middle or in between A and C. Not middle, between A and C. Right? So this way we got to know that we got to know that what is the location of the gene, what is the order. And even we can find out that A to B is 5 mu distance apart and B to C is 10 mu distance apart. And the basis, like you can construct a genetic map by performing a test cross. So here I'm showing you a 3-point test cross. So 3-point means that when you are considering loci of 3 genes in an organism genome. So here 3 genes are A, B, and C. And test cross, I'm sure you know what is test cross. When an individual heterozygous for 3 mutation, we are talking about 3-point test cross. So an individual heterozygous for 3 mutations is crossed with a homozygous recessive individual, triple homozygous recessive individual. Right? When you're crossing the heterozygous with the homozygous recessive parent, we call it as a test cross. So by using 3-point test cross, we can determine the phenotypes of the progeny can be scored. Now, when you are considering 3-point test cross, you have to consider 4 cases. Like I told you that the basis of recombination is crossing over. So there is a possibility that when no crossing over occurs, or there is a possibility that crossing over occurs between the genes. Now, because we are considering 3 genes, so crossing over can occur between A and B. Another case would be crossing over can occur between B and C. And 4th case will be when the crossing over occurs between both A and B and B and C. Okay, habitat, learn habitat, you are saying video is not clear. So just change your settings. You can change the setting to high quality. Okay, even on the screen you can see the instruction is mentioned how you can check, how you can change the video quality. Okay. So if you're not able to, just go to the settings and then you can click over on the quality and you can select 1080p. Okay. So yes, coming back to 3-point test cross. So first case is when you're considering cross between A and B. So here you can see we have 4 strands, 1, 2, 3, and 4, right? So crossing over is occurring between 2 and 3, right? So what will happen? There will be no change in first strand and fourth strand. You can see as it is. So we will get this combination. However, here, because of crossing over, this portion will come over here and this portion of the chromosome will come over here. So you have the recombinance which will be formed here. However, when you're talking about crossing over between gene B and C, then what will happen? You can see that again, there is no change in first and fourth, but you can see that the recessive allele C comes over this chromosome, the blue part you can see on red, and the red one comes over blue, right? So with the color, you can easily make out which part is being broken, and then like we have the shuffling of the genes which has occurred because of crossing over. And when there is a double crossover, that is crossing over occurring between A and B and C, then always remember, in case of double crossover, the middle gene is always get altered. So I'm specifically highlighting the important points which are very, very important and which are important for you to revise thoroughly. So here is a flow chart that you always have to follow when there is a question on the genetic mapping. So 3-point test cross helps you determine gene order and also helps you determine the distance between the genes. So whenever a question is about the gene order and the distance between the genes, this is the flow chart that you all always have to follow. Whatever data is given to you, like in the question, some data is given to us, right? Some progeny number is given to us. Identify which is double crossover and which is parental combination. Now, always remember, parental combination are the most common types. You will find them the highest number progeny to be the non-recombinant or parental, having parental combination, and the least common will be the double crossovers. Then you have to compare any one double crossover and any one parental to find the middle gene. Once you find the middle gene, if the order is given in the question is correct, it's fine. But if it is given is not correct, the order is not correct, you have to rearrange the sequence. This is very, very important. If you don't do this, then the rest of the steps will be wrong. So rearrange the sequences according to middle gene. Then find out single crossover event, calculate recombination frequency. Find out second single crossover, calculate recombination frequency. And lastly, plot the map with the frequencies. This flow chart, remember. So to make you recall and revise the concept faster, I have specifically made this flow chart. So make sure that you revise it thoroughly. So please go through it, because next I will be moving over to the question and then we'll be solving the question. So don't you think, students, that if you study this way, it's so interesting and so easy to remember the concepts? So make sure whenever you study, okay, see here at Biotechnica, we don't only help you in solving the questions, but even we tell you the strategies and also the methods of learning the concepts faster and in more easier and more enjoyable way, right? So just imagine you are reading the same thing line by line in the book or somewhere else in any other study material. This is much easy to revise and quickly recall, right? Okay, coming back to the question. So basically, just a minute. So basically, this is the data given to us, right? So we have to determine the gene order and we have to determine the distance between the genes. So what you are going to do is first identify, according to the flow chart I showed you, we have to first identify parental combinations, right? So which is the parental combination here? Quickly tell me. Most common one is the parental combinations, right? So here most common progeny is 350 and 300. So this is parental combination. Yes, Sharda, very good. 300, 350 and 300. Saptala, very good. Anupam, yes. Red, very good. Sakina, absolutely right. Krishna, Shrivani, Neha, correct. Yes, Krishna, Lakshmi, very good. So parental and least common are these two. So these must be the double crossovers. So according to the flowchart that I gave you, the steps I gave you, the first is we have to identify parental and double crossover. That part we have done. Now you have to find the gene order, whether the gene order given is correct or not. So I told you, you can consider any parental. Chosen this one, and let's say this one. Now we have to find odd one out. So if you look here, this is wild type allele, this is wild type allele on both the chromosome. This is wild type allele, this is wild type allele. This is wild type allele, this is recessive allele. So this one is odd one out, mutant allele, this is odd one out. So I told you odd one out will be in the center. So that means the correct gene order is H, ST and C. So the next thing is you have to rearrange the sequence, right? Rearrange the sequence in the data table given to you according to middle gene, right? Then find the first single crossover, calculate recombination frequency. Find second single crossover, calculate recombination frequency and plot the map with the frequencies. So here, when you rearrange, so I have rearranged the sequence. So ST will be in the middle, right? Now the next thing is you have to determine first crossover. That is single crossover event, that is crossing over between H and ST. See, H and ST, I told you 4 cases are there. When there is no crossing over, then when the crossing over is between H and ST, we took the example of ABC, right? So A and B. So here H and ST is given. Then ST and C. This is single crossover 2, right? So this is single crossover event 1. This is single crossover event 2. And double crossover will be when the crossing over will be between both H and ST and ST and C. So it is already given 4 and 6 over here, right? Now, so let me write the alleles. So I'm writing parental combination, okay? For single crossover, write down parental combination. So this one is parental and this one is parental, right? So H plus, ST plus, C plus, and here another parent. So when the single crossover event occurs, what will happen? The crossing over will happen then between H and ST. So what will be the combinations you get? The crossing over between these two. So this allele will come here and this allele will come here, right? So we should get a combination H, ST plus, C plus, and here we should get H plus, ST and C. So where this combination is given, you can see H plus, ST. See, this is one combination and another is this H, ST plus, C plus, right? So numbers are 86 and 114. So calculate the recombination frequency. So what is the formula for recombination frequency? Number of recombinants by total progeny into 100, right? So 86 plus 114 And even you will include double crossovers. Why? Because in double crossover, this is double crossover, one of the crossover is the single crossover. So you have to include double crossovers also. Then total progeny, when you add them all, you will get 1000 and then multiply with 100, so you will get 21 centimorgan. The remaining two, 90 and the 50, will be the second crossover event. That is the crossing over between ST and C, right? And when you solve it, you will get 15 centimorgan. So that means answer here will be option 3. Okay, Dana Priya, which part you want me to explain? See, you just have to follow the flow chart, okay? Whatever flow chart I told you, just follow that. Single, when you see, to determine the single crossover event, you have these. This is one parental combination. It already given, okay? This is your parental. I've already told you in the beginning, most common are the parentals. So when I say crossing over occurring between H and ST, this is single crossover event 1. So this results into the crossing over will results into what? Shuffling of the alleles, right? So you should get here So here you should get H, ST plus and C plus because this one will move over here and this one will move over here, right? And here it will be H plus, ST and C. So I have just seen where these combination is mentioned. So one is over here, another is over here, right? This one is over here and this one is over here. So rest two will be what? Even you can check what if the crossing over is between ST and C. When crossing over is between ST and C, what will happen? This one will move over here and this one will move over here. C allele will move, right? So H plus, ST plus, and you should get C. So this is the combination I'm getting here. And another H, ST, and C plus over here I will get. Right? So this is the combination. Clear to everyone? Okay. Now let's move over to another question. So without wasting the time, let's quickly solve the questions. Now this question is about the linkage. This question is about tetrad analysis. Linkage, I have another question. So tell me, so far, was it helpful? Did you learn the concepts? Did you revise? Were you able to recall the concepts? Please tell me yes or no in the chat. Anupam, yes, previous year question. The question I showed you earlier, it was the previous year question only. 2021, I think it was. Okay, okay. Thank you so much for your response. I'm really glad to know that it is really helpful for you all. Okay, okay. Thank you, everyone. Thank you. So go through the question. Tell me what is the answer here. Okay, Rumesa, flowchart was helpful. That's great. Good to know. Thank you for letting me know that it was really helpful to you. The following table summarizes the result of a cross between two strains of Neurospora, having the alleles, dominant allele with D and recessive allele with lowercase letter d respectively. The table shows the different pattern of octard arrangement and number of ascus observed of each type. Based on the above, fill in the blanks from the options given below. So the first two columns are from meiosis with no crossover between locus D and So A means that is you have to see whether D allele will come or centromere will come. That is between locus D and D allele or centromere. Then the patterns from these two columns represents dash segregation pattern. That is first division or second division. And the distance between the locus D and the centromere is C map unit. So in the options given is 10 or 20. So what do you think is the correct answer here? So from genetic mapping, either they can give the questions like the one I showed you, the previous question I showed you, or based on tetrad analysis. Okay, Shrivani, you are asking that, ma'am, whether the same question repeats. So, Shrivani, not the same question repeat. Maybe they change the names, maybe of the organism or the data values which are there. Okay, so it's not the exact same question you will get, but similar type of question you can get in the exam. I hope this answers your question, Shrivani. So, yes, what is the answer here? Tell me. You have to keep a record of time also. Don't take too much time. Okay, so Maria, you have no idea about tetrad analysis. Don't worry, it's okay. See, students, if you have not have left anything, okay, if let's say you have studied genetic mapping, but you have left tetrad analysis. So I'll suggest that although I will be explaining the concept to you, but don't focus on that in like more in detail, because I will suggest that don't start anything new at this point. Whatever you have studied so far, just focus on that and revise that thoroughly. Okay, because see, there are many, each of you have your own way of studying. Each of you have selected the different units, right? Like selection of the units will be different for you all, right? So just focus on whatever you have, whichever units you have selected, whichever topics are important from those units, just focus on that. Okay. But I will be explaining you, Somaria. Don't worry for the like tetrad analysis. You should know what basically it is all about. Okay. So Kavya read, you are saying option B. And just guessing, Arzu. Okay. But don't, in the exam, don't do the guesswork. Okay. If you're not sure, then leave that question. Don't do the guesswork in the exam, because there is a negative marking, and majorly the students, they lose their marks in negative marking. So don't do the guesswork in the exam. Okay. So let me just quickly tell you the concept behind this question. So basically this is about the Neurospora. So actually when you talk about the fungi, like Ascomycete, when we talk about, so when the, like, in the fungi there is no, to differentiate between two different strains, we are representing them with the, like, the fungi of one mating type. When the cell of one mating type fuses with the cell of another mating type, the zygote form will be diploid. Okay, so when you talk about the life cycle of Neurospora, so Ascomycete, it belongs to Ascomycetes. So the mating, one cell of one mating type when fused with another, so it's a mode of asexual reproduction. Sexual reproduction. So basically here you can see that the F1, like the zygote, will be heterozygous, and then further the meiotic and mitotic divisions will occur. Now in Ascomycete fungi, the ascospores are arranged in a cylindrical bag-like structure. The cylindrical shaped structure, we call it as ascus. So basically what happens that when meiotic division, meiosis 1, meiosis 2 will occur. So it results into formation of 4 ascospores. We call it as tetrad. And when the meiosis is followed by mitosis, it results into formation of a double tetrad, or you call it as octad. So in the octad, you have 8 ascospores which are there. Now 4 are of one type and 4 are of another type. So basically 4 will be of one type and 4 will be of another type. Okay, here 2 will be of one type, 2 will be of another type. Like this way. So basically, you have to remember the examples. When we talk about arrangement of the ascospores in an ascus, if they are orderly arranged, like here you can see this is an ascus, the cylindrical bag where the ascospores arranged are very compactly arranged, right? So basically there could be no shuffling of the ascospores which can occur, right? No shuffling of ascospores occur. So this is ordered arrangement. But if I draw the, like if this kind of arrangement will be there, right? So you can see that there are a lot of spaces, they are not compactly arranged and shuffling of the ascospores can occur. So this is unordered arrangement. So when we talk about the tetrad analysis, we have to consider two cases for ordered tetrad and for unordered. Now from exam point of view, you have to remember the example. The example for ordered tetrad is Neurospora. Now Neurospora always have an ordered tetrad arrangement or octad arrangement. Whereas in yeast, like Saccharomyces cerevisiae, there you have the unordered arrangement. So first of all, you have to remember the example. Now, when you talk about ordered tetrad, you have to consider two cases. Okay. So this is again, you can remember in terms of flowchart that when you talk about ordered tetrad, two cases are there. One, when there is no crossing over, and second, when there is crossover. Now remember, when there is a crossing over, when there is no crossing over, the segregation of the chromosome occurs in the meiosis 1. So here we are, instead of using D and these two alleles, A and A alleles we have written. This diagram is showing explaining it with the A allele. So basically during the first meiotic division, so when no crossing over, we say the segregational pattern is meiosis 1, and you can see that finally meiosis 2. So basically here you can see these two chromatids will separate, right? And then further, if mitosis occur, then octad. So 4 are of one type, 4 are of another type, right? So based on the arrangement, if this allele is present here and this allele is present here, so you will have this arrangement, 50-50. So here, what is the ratio? 2:2. Like in Mendelian principles, Mendelian experiments, we have studied monohybrid, cross phenotypic or genotypic ratio, right? Similarly, here you have to remember the ratio of the ascospores, type of ascospores arrangement in the octad or tetrad. So it is 2:2, like 2 are of one type and 2 are of another type, or 4:4 in octad. So 4 are of one type and 4 are of another type. So this is what is shown here. And these are similar to parental combination, right? There is no recombination here because no crossing over. However, what another point you have to remember is that when there is a crossing over, so crossing over to occur for the allele A, like you have to consider one reference point from where the crossing over is occurring. So that is centromere. So when the crossing over between locus A and the centromere occurs, so you can see depending upon which chromatids or which strand is participating in the crossing over, accordingly we'll get the you will get the recombinants. So like here you can see first and fourth strand is not participating in crossing over, but second and third is participating. So can you see some brown on yellow comes, right? Or yellow on brown comes. So this is with the help of color you can easily identify, right? But the point is that this allele moves over here and this allele moves over here. So you can see these two are recombinants and these two are similar to parental combination, right? Now here, when the crossing over occurs, remember it is M2 segregation pattern. That is, the chromosomes will segregate in meiosis 2 and ratio is 2:2:2:2 or 2:4:2. Now, how these ratios will occur? Depending upon the crossing over is occurring between two chromatids, which two strands, okay? Which two strands, accordingly you'll get the ratio. So like here, the crossing over is between 2 and 3. So you get 2 of one type, 2 of another, right? So 2:2:2:2. But please note that this one and this one is not same. Okay, one will be recombinant. Like this one is similar to parental, but this one is recombinant. Okay, so don't confuse that these two are same. No, one will be parental, another will be recombinant. Here also one will be recombinant, another will be parental. So this is just an arrangement. That arrangement will be of the ascospores will be like this, or Like for example, when the crossing over is occurring between second strand and fourth strand. So the ratio is 2:4:2. Okay, so ratios you have to remember. So in an octad, the ratio is 2:2:2:2 or 2:4:2. So each particular pattern is equally frequent as any other M2 pattern. It's just that which two, the crossing over is occurring between which two strands. So this is about ordered tetrad. But when you talk about unordered tetrad, the example is of Saccharomyces cerevisiae. And to understand the unordered tetrad, we considered the two pair of alleles to understand the unordered tetrad. So here you can see one parent is wild type, having the wild type alleles for both A and B gene. But parent 2 is a double mutant, that is having mutant allele for A and B gene. So if the progeny, like if these two are crossed, the progeny will be double heterozygous, right? And again here, depending upon whether crossing over is occurring or no crossing over is occurring, three types of tetrads can be formed. One is parental ditype, another is tetratype, and third is non-parental ditype. Please note students, here I can only focus on the key points here. I cannot, like we have a complete detail about the tetrad analysis, but because since we are revising, I can, like we can revise the key points here, okay? So here parental ditype means that all four will be parental, where two will be of one type and two will be of another type. However, in tetratype, you have one parental, like all four different. This is parental resembling to this parent, parent 1, this parental resembling to parent 2, and recombinants, you can see these two are different. So all four different types, tetratype. And in non-parental ditype, you will get all four recombinants. So non-parental means recombinant. And ditype means that two will be same and these two will be same. Okay, so these three will be formed. So in any question based on tetrad analysis, first we have to identify whether the question is based on ordered tetrad analysis or unordered. And if the question is about recombination frequency, the formula used for ordered and unordered is different. For ordered tetrad, the recombination frequency formula we have studied, it is number of recombinants by total progeny into 100, right? So here for ordered tetrad, it is half into M2 SK by total SK into 100. So why half? Because you know that half are recombinants and half are parental. So this is the reason, because the recombinants are half of M2 SK. So this is the reason we are adding half here by total SK. But if the question is about unordered tetrad, the formula is half the number of tetratype plus number of NPD, non-parental ditype, because in NPD all are recombinants. However, in tetratype, these two are parental and two are recombinants, right? So again, half. We are writing half here by total number of SK into 100. So I'm sure those who have studied tetrad analysis, you are able to recall and revise the concept of tetrad analysis now, right? So let's go back to the question. So this was the question. Can you please quickly solve it? The following table summarizes the result of a cross between two strains of Neurospora having the two alleles. The table shows different pattern of octad arrangement and number of ascus. So first of all, tell me, is this a question of ordered tetrad or unordered tetrad? How will you find out? Which organism is given here? Is it ordered or unordered? Ordered. Very good, Sunshine, Sakina, Lakshmi. Yes, very good, everyone. Ordered, because East is an example of unordered. So this question is about ordered. And I told you for ordered, we have to consider two cases. No crossover and crossing over. Then what what happens in no crossing over? Which segregation pattern? M1. For crossing over, M2. Then second, you have to recall ratio. For no crossing over, the ratio is 4:4 in an octad. And in M2, it is 2:2:2:2:2 or 2:4:2. So the table is given to you. Just consider which is no crossing over, which is crossing over. So it is mentioned first two columns are from meiosis with no crossover between locus D and centromere or another allele. So these are the first two columns which you have to see. What is the ratio here? So by going through the ratio, you can find out it's a case of crossing over or no crossing over. Yes, quickly tell me. What is the ratio? These four are of one type, these four are of one type. These four are of one type, these four are of one type. What is the ratio? Very good, Soumya, no crossover. So means 4:4 ratio. Right? So when there is no crossing over, yes, 4:4, Kavya, Krishna, Swagata, Shweta, absolutely right, Lakshmi, correct. Danpriya, very good. Revti, yes, correct. So that means the locus D and centromere it will be, right? And then let's see the 3, 3rd, 4th, 5th, and 6th column. What is the ratio? So first division here means meiosis 1 and second division here means meiosis 2. So by seeing the ratio, if it is a case of no crossover, M1 segregation pattern. If it's a case of crossing over, then M2 segregation pattern, right? So if you are saying centromere, our answer cannot be A and C. Answer will be between B and D. So whether it is a first division or second division segregation, what is the ratio? These two one type, these two one type, these two, these two. So 2:2:2:2, right? And here again 2:4:2, right? So this occurs when there is a case of crossing over, right? Here also 2:4:2. So M2 or M1. Now this pattern that you have to determine for the first two columns are what? This B is for first two columns. And then this C part you have to see to determine the distance. So what is the answer here? Okay, some are saying option 2, some are saying option 4. So first of all, you have to always look for ordered, okay, or unordered. What is mentioned, okay? You have to always see that by seeing the example of the organism, whether it's a case of ordered or unordered. Now, from the example, it is Neurospora, so it's an ordered arrangement. Then ordered tetrad analysis is done by finding distance between gene locus and centromere. So A will be centromere. So that means answer cannot be option 1 and option C, right? Option A and option C. Then I told you that if first two columns, if you see the arrangement is 4:4, that is no crossing over. And no crossing over means first division segregation, right? So by seeing this only, you can make out that answer should be option B. But again, to confirm, like 3, 4, 5, and 6, it is crossing over which is mentioned because ratio is 2:2:2 or 2:4:2. Now, what is the formula for calculating the distance? I told you genetic maps are based on recombination frequency. So number of recombinants by total progeny into 100. So here the recombinants are calculated as half into M2SK. That is the formula, right? So M2SK are what? 14, 16, 17 and 13, right? So you add them up, divide by 2, and then so half into, when you sum them all, divided by 300, that is total progeny into 100, you should get 10 centimorgan. So answer here is option B. Clear to everyone? Now last question, we'll do this one and then we'll move over to unit 13. Okay. So this is about linkage. So double haploids are plants derived from single immature pollen and doubled artificially to form diploids. A double haploid, okay, DH is double haploid population was created from F1 progeny derived from a cross between two parents P1 and P2. One resistant R and other sensitive to white rust. The parents F1 and DH population were profiled with set of codominant marker which is represented below. So codominant marker means which is able to differentiate between homozygous and heterozygous condition. And then a table is also given where the percentage, proposed percentage of 4 different types are shown. These 4 different types are shown. Okay, these are 1, 2, 3, 4. Assuming the marker is unlinked or it is linked. So basically, what is linkage? Linkage, it is considered to be a deviation from independent Mendelian principle of independent assortment. Linkage means that the marker, as we are talking about a marker gene, so that means linkage, it means that genes are always inherited together. There will be chances of them to undergo crossing over will be very less. So whether the marker is associated with the gene, if it is linked, that means it will always be inherited together. So the concept here is always remember when you talk about independent assortment, right? Like for example, when you see here, this is a independent Mendel's dihybrid cross, right? So we know that when yellow round plant with round yellow seed is crossed with wrinkled green, the progeny will be all round yellow, right? And on selfing of F1 generation, you will get the phenotypic ratio 9:3:3:1, where 9 will be showing the dominant trait that is round and yellow. 1 out of 16 will be showing the recessive trait that is wrinkled and green. But you can see some new combinations are also formed which were not there in parent generation. So the basis of recombination I told you is always crossing over. If no crossing over, like here A and B genes are alleles are there. If no crossing over, the gametes form will all have the parental combination. They will be non-recombinant. However, if the crossing over occurs, like here the crossing over is occurring between second and third strand, right? So you can see that some combinations are parental, non-recombinant ones, but some are recombinant. So because crossing over has occurred between these two, the gametes will have the recombinant combination, right? So here half will be recombinant, half will be non-recombinant. So in independent assortment, when you talk about crossing over leads to recombination. However, when you talk about linkage, linkage is deviation from the Mendelian principle of independent assortment. So linkage again can be of two types, complete or incomplete. Complete linkage means no crossing over, this case. So the gametes form will be all parental combination. But it is observed. See, genetics is all about, variations, exceptions. So crossing over can occur, sometimes can occur because linkage is rarely complete. Now question arises when there is no crossing over, when there is a complete linkage. Linkage means that the two genes, let's say A and B are linked. That means they will always be inherited together. So no crossing over, gametes form will be all non-recombinant. But if it is a case of independent assortment or incomplete linkage, how we will differentiate? So the answer is we always test for linkage with a test cross, where you cross a heterozygous with a homozygous recessive parent. Now, can you tell me what is the dihybrid test cross ratio? Mendel's dihybrid test cross ratio? Quickly tell me what is the Mendel's dihybrid test cross ratio? Yes, what it will be? Dihybrid, Kavya, I'm asking. You're telling me about monohybrid. So you are right. But for dihybrid, it will be 1:1:1:1. Neha, absolutely right. So all the 4 progeny in equal proportions, where 2 will be parental type, 2 will be recombinant. So gametes formed in independent assortment will be parental type and recombinant type in equal proportion. But in incomplete linkage, parental type will be more always and recombinant type will be less. So these are again the concepts that you have to recall or revise for the questions based on linkage. So moving back to the question. So the question is that this table is given to you and this gel profile is given to you, and you have to find out that which of the following options correctly represent expected ratio for unlinked and linked respectively. So what do you think is the correct answer here? Option 1, 2, 3 or 4? Okay, Lakshmi, you are saying option 2. Okay, what about others? Okay, dream narrator, you are also saying option B. Fine. See, this is parent 1 generation, this is parent 1, parent 2, and this is F1 generation, right? And these are all the progenies, right? So 1, 2, 3, and 4. So you can see these 2 are resistant and these 2 are sensitive. So first of all, you have to find out that if they are unlinked, then which one option is correct? Is it A or is it B? So unlinked means what? What I told you, unlinked means there is no linkage. So what do you think? Some ratio is given to you, right? So what do you think? All the progeny will be, if they are unlinked, then they should occur in equal proportion, right? 1:1:1:1. So that means out of A and B, A is correct, right? So when I say A is correct, that means answer cannot be option 3. Now we have to find out that A is 1, 2 or 3, right? So now the condition given is linked, right? So in case of linkage, right, I told you complete linkage, no crossing over, no recombinant. So here it is a case of incomplete linkage, where along with parental you will get recombinants also. So one concept I told you, that parental will always be more common and the recombinant will be less, right? But here 4 progeny, 1, 2, 3, 4. So you have to find out which one is similar to parental and which one is recombinant. So for this, we have to study this gel profile. So here you can see this is R and this is R. So they are having the same pattern, right? So this means this is similar to parental, having parental combination. But if you compare this one with this one, don't you think this is recombinant? Because this one doesn't match with the R1. It matches with S1. But we have to compare R with R, right? Then if you see S, so that means second is recombinant, right? Second is recombinant. First is parental, second is recombinant. Then third, if you compare, this should resemble to parent 2. But you can see the pattern is different. So again, this is recombinant. However, fourth one is similar to parent 2, S with S. So that means 2 and 3 are recombinant, right? Now you have to calculate this, what is the distance. The question in the question, it is given linked at a distance of 10 centimorgan, right? So that means for this, so first I have considered unlinked. So when the gene and marker are not linked, equal probability of producing all possible combination. So that is why A is correct. But when they are linked, we have to determine which is parental, which is recombinant, right? So I told you lane 1 and lane 4 are parental and lane 2 and 3 are recombinants, right? Now distance given is 10 centimorgan. So because each, the second and third are recombinant and the distance is 10 centimorgan, so each recombinant proportion will be 10 by 2, that is 5%. And each parental proportion will be, so total progeny, if you sum them all, it is 100. So 100 minus 10 by 2, that gives you 90. And for each parental, it will be 45%. So this is the reason for linked A, the 1 is correct. So answer here will be option 2. So yes, Sakina, Dream Narrator, Soumya, you all have answered. Sunshine, your answer is absolutely right. So now moving over to unit 13. So genetics questions we have discussed, and we have recalled, revised so many concepts, right? Based on probability, based on tetrad analysis, based on gene mapping, based on linkage. Now moving over to the unit 13. So here are some questions on your screen. So this is the question based on restriction digestion. Please go through it and tell me the answer here. So given below are names and recognition sequences of a few restriction enzymes that are used for cloning experiments. The cleavage site of each enzyme is indicated, and given below are different vectors. Column A and insert column B generated by digestion using above enzyme. So the sequence of the enzymes are given here. Which one of the following options represent the correct combinations of vector and insert respectively, which generate compatible ANDs for ligation? So always remember, the trick here is that you always have to choose the combination of enzymes which can generate compatible ends. Like vector can only bind to a gene of interest if they have the compatible ends. So yes, quickly tell me what is the answer here. Keep the record of timing also. You have to check in how much time you are able to solve the question. So everyone should try. Okay, majority says option 2. So it's great you are able to solve the question quickly. I really appreciate you all for this. Very good. That means you all are really practicing the questions. Okay, majority. For this, I am getting the answer. Option 2, majority says option 2. Option B. Okay. So basically, this question is about restriction enzymes, right? So the concept here is, like I'm sure you know, that restriction enzymes, also called as restriction endonucleases or molecular scissors, and the important, like why these restriction enzymes are so important in gene cloning and recombinant DNA technology, because they don't cut randomly. They recognize a specific sequence and makes a cut over here. Now, the cut produced by restriction enzyme can be either blunt or sticky, that is cohesive. So here you can see EcoR1 is a restriction endonuclease. It recognizes GAA-TTC sequence and cuts within the sequence, right? Now, when the enzyme recognize, like some enzymes generate blunt ends like ALU1. So basically, this is the recognition sequence for ALU1 AGCT. So when it recognizes the sequence and just cut in middle of the recognition sequence, the ends generated are blunt end. However, EcoR1, it generates sticky ends or cohesive ends. So cohesive ends, basically, why they are cohesive? Because they can stick back by transient base pairing. A can pair with T can pair with A. Okay. So here in sticky ends, basically, the cut is not made in the middle of the recognition sequence. It's the staggered cut. That is, the enzyme will cut on one strand over here and on another strand, 2 to 3 or more nucleotide away from the first cut where it is made. So like here. So this results into the overhang region. Like this part, you can see it is single stranded, right? And why they are sticky end or cohesive end? Because they can stick back by transient base pair. So whenever we are making a recombinant DNA molecule, any blunt end could be compatible with any blunt end because they have to be joined with ligase enzyme. But for the strong joining, the sticky ends are preferred. So that means when you're cutting the vector, you have to cut with enzyme which generates sticky end. And also either you can use the same enzyme for cutting the gene so that the ends generated are compatible, or the combination of the enzymes which can generate same type of sticky end. So that you can use. Now in the question, we have been given the sequences and we have given the vector and insert fragment, right? So here, if we see option 1, A is 2. So that means EcoR1. So EcoR1 generates sticky end. And you can see 2, it is HINC2. So HINC2 generate blunt end, right? So one is sticky end cutter, another is blunt end. So that means this cannot be the answer, right? So this combination of A2 is not possible because one generates sticky end, another generates blunt end. So when I say, so A2 is not possible, that means this cannot be the answer. Now, the next we can check for B2. So B here is EcoR5, which generates the blunt end. And 5 is, second is HINC2. So yes, HINC2 generate the blunt end. So I told you any blunt end could be compatible with any end. So yes, this combination is possible, right? So we can further confirm by taking E and 1. So E is HINC2 and 1 is EcoR5. So HINC2 and this is EcoR5. So yes, this is also possible. So when I say E1 is possible, that means E3 and E2 is not the answer. So it's very direct answer here is option 2. Excuse me. But you can further confirm. So you can confirm D and 4. So just check, arrange the sequence and check whether it is compatible or not. So D here is BG3. Now please note whatever sequence given to you here is, it is always 5' to 3'. Okay. So BGL3, so this is the sequence, right? So always remember this is 5' to 3'. And don't forget that when you talk about gene, it is double stranded, right? So if this is 5' to 3', you have another sequence that is 3' to 5'. So here it should be T, it should be C, here it should be T, here it should be A, here it should be G, and here it should be A, right? So that means if BAMH1 sequence 5' to 3' is given, that means this, if we are looking for compatible ends, that means this 3' to 5' should be compatible with this 5' to 3' BAMH1 sequence or vice versa, right? So here you can see, if this is the sequence, you can see G is compatible with C, A is T, T with A, and C with G. So at least 4 nucleotides are compatible. So yes, this could be the correct combination. And this is the reason the answer here will be option 2. Okay, so don't waste time in whatever table is giving to you. So arranging, like just comparing all of them. No, just see the options and try to omit options, like 2 options, start with 2 options, omitting 2 options out of 4, and the rest 2 which are remaining, you can compare to find the answer. Because if you start comparing all of them all together, it will be so time consuming. Okay. Now moving on to another question based on flow cytometry. A mixed cell population was stained with 2 antibody, one specific for cell surface antigen A and other specific for cell surface antigen B. So that means there is a cell having antigen A and another cell having antigen B. So obviously to detect antigen A, we are using anti-A antibody, and for B, anti-B antibody. Now anti-A was labeled with fluorescein and anti-B was labeled with rhodamine. Then you are analyzing the antigens, presence of antigens by flow cytometry. So which of the following is the correct outcome for this cell population? So here you have to recall the concept of flow cytometry. What is flow cytometry? What is its application? And also about the sorting of the cells. So FACS, fluorescence activated cell sorting, you have to recall here. Okay, Neha, Soumya, you are seeing option C. What about others? Okay, Arzu, option C. Shweta, you're also saying option C. Okay, Krishna, option C. Sandeep, you're saying option A. Okay. Okay, some are saying option A as well. Okay. So basically here you have to recall the concept of flow cytometry. So flow cytometry is what? It's a powerful tool used by immunologists to study the multiple parameters on the cell. Multiple parameters means, first of all, to identify the cell on the basis of the specific antigens present on the cell surface. We call them as markers, right? Like certain markers, certain antigens which are specific to a particular cell. So we can identify that particular cell based on the antigen present on its cell surface. Secondly, you can analyze multiple parameters, parameters like what is the size of the cell and the granularity or complexity. Like for example, when you talk about white blood cells, we have agranulocytes and granulocytes, right? So immunologists, when you have a mixture of the cells, they can find out which are granulocytes, which are agranulocytes. So agranulocytes, you have lymphocytes, monocytes, which don't have granules, and granulocytes like eosinophils, basophils, right? So cell population can be characterized. You can identify a particular cell based on antigen present on its surface as well as intracellular. So the flow cytometry, it's a quantitative technique. Even if you have a cell population, mixture of cell population, you can find out how many of the cells are B cells, T cells, how many are natural killer cells, how many are lymphocytes. So it quantifies the frequencies of cells. And actually, the basis of the flow cytometry, the principle of flow cytometry is immunofluorescence. To detect a particular antigen, you use antibody. So immunofluorescence, immuno prefix means when you use antibody to detect an antigen. Okay. And fluorescence, because these antibodies are labeled with a fluorescent dyes. So let's say in our question, like A and B antigen are there. So one antigen, antibody you label with one dye, another with another dye. So like here also you can see we have a mixture of cells, right? Some cells are having only A antigen, some are having B antigen, right? So here like green one having A antigen and red one representing the B antigen, right? So red one is representing B antigen. So basically the antibody, anti-B antibody is labeled with a dye which emit red fluorescence, and anti-A antibody labeled with a dye which emit green fluorescence. Now this is a mixture of cells. Okay, you can see some cells they don't have A or B antigen, some cells having both A and B antigen. So we have four type of cell population. One is having no A or B antigen. So I can represent it like this. So A minus B minus. B here minus doesn't mean charge. It means absence of A and B antigen on cell. Second, presence of A but lack of B antigen. Third case will be where A antigen is not there but only B antigen is there. That is this one, the red one you can see here. And fourth is where you will have the cells having both A and B antigen. So first of all, to the cells you add anti-A and anti-B antibody. Depending upon whatever antigens they have, antibodies will bind. And these antibodies are labeled with the dyes, different fluorescent dyes. Now, you add it into the flow cytometer. So in the flow cytometer, there is an arrangement where the cells pass as a single file. They arrange as a single file and they pass one at a time. So we have a sheath fluid, that is a buffer saline solution. Along with it, the cells, they will pass as a single file. And as they pass, the laser beam will pass through it. So as the antibodies are labeled with a fluorescent dye, these fluorescents will absorb the light. The fluorescent dye will absorb the light and emit light of longer wavelength, which call as fluorescence. And this fluorescence will be detected by the detectors. But the advanced application of flow cytometry is fluorescence activated cell sorting, where even you can sort the cells into different containers or different vessels. So in FSCS, the only additional part is you add a deflection plate. So based on which fluorescent dye you have used, accordingly the charges will be there on the cell. And you can see that the cells lacking A and B antigen will not be deflected as they don't have any antigen and hence no dye conjugated antibody. But here those cells are collected which have both A and B antigen. Here the cells having either A or B antigen will be detected, right? So basically those cells having positive charge will be deflected towards negative charge plate. Those cells having positive charge, negative charge will be deflected towards positive charge plate. Now this is just a working principle of flow cytometer which I have shown you here. But from exam point of view, you have to remember this dot plot. So this is how the data analysis of flow cytometer is done. So what you have to do is always remember the trick here is just draw the square, divide it into 4 quadrants. Remember what you are labeling on X and Y axis. Accordingly, you will write these combinations. So here anti-A antibody is on X axis. That means here those cells having A antigen will be detected, right? And here those cells having B antigen will be detected. So always remember Like if you consider it as a like in a number scale, we write down like number scale, we have a 0. So any values of more than 0 will be positive, and any values less than 0 will be negative, right? Same here, you have to remember. So I'm giving you a trick here to remember, okay? So when I say A antigen on x-axis and B antigen on y-axis, so here, like if this is the starting point, so that means this quadrant represents presence of A and this indicate absence of A. This indicate absence of B, this indicates presence of B. So here I can write down the combination, these combinations are there. So that means this quadrant represents absence of A and B antigen. This quadrant represents presence of A antigen but absence of B antigen. This quadrant represents presence of both A and B antigen. And this represents absence of A but presence of B antigen. Right? Because absence of A but presence of B antigen here. Right? So this is called as double negative. This is double negative and this is double positive. So if you just remember this, based on this, the questions are asked. Now, in the exam, they can give you any marker, they can give you any fluorescent dye, anything they can mention it to you. Okay. So always remember here in the question, the question is based on the same concept. So 4 data are given to us, right? 4 dot plots are given to us. So I told you out of 4, always remember this one will be double negative and this one will always be double positive. So first thing you can check is for double negative and double positive. So here, this is wrong because double negative should be here. So yes, in B, double negative is here. Then check for double positive. It is not there. That means B cannot be the answer. If you see C, here double negative, double positive. Here also double negative, double positive. That means answer is between C and D. We can omit 2 options out of 4. Now I told you because the green fluorescence. So in the question, we have labeled the green fluorescence will be given by fluorescent and rhodamine gives the red fluorescence. R for rhodamine, R for red. And B antigen we have labeled with anti-B antibody. We have used anti-B antibody for B antigen conjugated with rhodamine. So red fluorescence here is for B antigen, right? Detects the presence of cell having B antigen. So this should be B plus and absence of A, right? But you can see the same thing is given over here. So this cannot be the answer. If you look here, absence of A, this should indicate absence of A, but presence of B, right? And here, this should indicate presence of A and absence of B. So yes, this is what is given here. So answer here is option 3. Easy, right? If you just remember this trick, don't you think you can solve this question? Yes or no? Quickly tell me. Was it helpful? Yes, was it helpful? Okay. Thank you, Rabula Arzu, for the response. Thank you, Somarya. Very good. Thank you for the response. Sri Lakshmi, Dilshad, Swagata, Shweta. Thank you very much, Krishna. Good to know that you like this concept, like this trick. Now moving over to another question. Now this is a little bit tricky question. This question is not direct. This question is based on combination of unit 1 and unit 13. So circular dichroism from unit 13 and protein secondary structure from unit 1. So if you are familiar with circular dichroism but not the secondary structure of the protein, you cannot solve this question. If you are familiar with secondary structure but not with dichroism, circular dichroism, you cannot solve this question. So that means to solve these questions, you need to be thorough with both the things. So first of all, what is circular dichroism? It's an absorption spectroscopy, right? So just go through the question, we'll revise the concept, and then we'll solve this question. So actually you have to compare all the 3 spectrum which is given to you. And also you have to go through the options one by one. and you have to omit the incorrect options and find the correct one. So circular dichroism in the far UV region informs about secondary structure. So this is already given to you. Information is already given to you that in circular dichroism spectroscopy, if you analyze the secondary structure of the protein, what are secondary structure of protein? Alpha helix, local arrangement, right? Alpha helix, beta, beta sheet, right? Beta turn. So far UV region informs about secondary structure, and near UV and tryptophan emission spectra informs about tertiary structure. So tryptophan is an aromatic amino acid, and it is basically being responsible for absorption of light. So circular dichroism, it is an absorption spectroscopy where we study about the absorption of the light by the matter or biomolecules. Now panels shown is intrinsic fluorescence emission spectra of protein X. This is far UV circular dichroism spectra and this is near UV. Now, even you can see three colors, black, red and green. So curve here represent the spectra of protein X at pH 7, which is shown with black line, pH 3 shown with green color, and pH 7 in presence of a denaturant, that is guanidine hydrochloride, with red. So what does the experiment report? Just think about it, how you are going to solve this question. So what do you think is the answer over here? So for this, I told you, you need to be familiar with both circular dichroism and secondary structure of proteins. And you should know how to read the graphs, how to read the spectra. Okay, Sandhya, you are saying option 1. Fine. What about others? So meanwhile, you answer. Let's quickly revise the circular dichroism spectroscopy. What it is? It's an absorption spectroscopy, right? So basically here you study the differential absorption of left and right circularly polarized light. So differential absorption we give by this following formula, like the differential absorption of left circularly polarized light minus differential absorption of right circularly polarized light. And basically circular dichroism is specifically used for chiral molecules. Chiral means which are asymmetric. Like for example, when you talk about the amino acids, so they have a chiral carbon, right? Why? Because they have all the 4 different groups attached, except glycine. If you see all other amino acids, they have side chain R, carboxyl group at one end, amino group at another end, and hydrogen atom. So they are asymmetric, right? And usually the circular dichroism spectrum we report in terms of like theta, ellipticity. So in the question, it is clearly mentioned that far UV, we use it to study about the secondary structure, and tertiary structures are used to study, tertiary structures can be studied by tryptophan emission and near UV. So here we will compare all the, like we'll go through all the four options one by one, okay? And we'll see which statement is correct or incorrect. We have to find the correct statement. So option one was that protein is fully folded at pH 7, acid-induced molten globule at pH 3, and unfolded in 6 molar guanidinium hydrochloride. So basically, if you look at the near UV, okay, so near UV tells you about the tertiary structure, right? So if you compare with the, like this black line, if you see, and this red line you see, and green line you see, right? So if you see the green line, which is at pH 3, showing at pH 3, and black line at 7, you can see that there's a clear change in near UV circular dichroism, right? And even emission spectra you can see. This is 327, this is 340, right? So this indicates that the pH 3, there must have been a change in tertiary structure, right? So specifically, you have to go through all the lines, right? So at pH 3, because you can see the green one is like this, and the red one, the black one is like this. So definitely from here to here, you can see that definitely there will be a change in tertiary structure. And even in molten globule state, when we talk about, the secondary structure can alter. Now, it is mentioned that unfolded, the protein is fully folded at 7, and there is an acid-induced molten globule at 3 and unfolded in guanidinium hydrochloride. So here, guanidine hydrochloride act as a denaturant, right? So definitely, if the protein is denatured, the tryptophan will be exposed. And if it is exposed, we should get the emission spectra for the tryptophan should increase. So yes, that has been indicated with this red one, the red spectrum. If you see, it is the emission spectra, right? And if you see the far UV, the red line is flat. Like if you look here, the black one, which is folded at pH 7, but at red line, it's completely straight. That means there is no absorption shown here. No absorption shown here means that the protein has lost its secondary structure and even tertiary structure, because far UV indicates the content of secondary structure, right? So yes, that means if it is now unfolded, right? So that means this statement is correct. However, if you see option 2, it is protein secondary structure is reduced at pH 7 and protein has formed beta fibrils at other two conditions. So secondary structure is revealed by far UV, right? So if you see here that if you see the black line, which is representing the pH 7, it remain unchanged, right? So it means, so that according to option 2, it is saying it is reduced and it has formed beta fibrils. So no, this is incorrect. Then if you see option 3, the statement is that change in fluorescence and near UV. Okay, the change in fluorescence, that means the spectrum, and the near UV indicate increase in hydrodynamic radius at pH 3 and 6 molar guanidine hydrochloride. Now, for what is hydrodynamic radius? Hydro means solvent, and dynamic, here we are talking about shape dynamic effect. So RH basically represents the hydrodynamic radius, which includes the solvent, that is hydro, and shape dynamic effect. So that is why hydrodynamic effect, like when the protein is in presence of certain solvent, right? So hydrodynamic radius, it includes the solvent and the shape dynamic effect. So theoretically, it is predicted by this. Okay, so basically hydrodynamic radius for native folded proteins predicted by this, and denatured state, it is this, right? So that means in denatured state, it should less, but here it is mentioned it should increase. So this is again wrong. This is incorrect statement, right? See, to solve these questions, your concepts need to be very, very thorough, very, very clear. Okay, you should know what these terms means. Okay, you should know what are secondary structure, what are tertiary structure. And if you see, again coming back to the question, if you see option 4, there is an extensive denaturation of protein both at pH 3 and 6 molar guanidine hydrochloride. So denaturation, if you see far UV, pH 3, which is represented with the like pH 3, it is with the green one, right? So yes, pH 3 is with the green, right? So here you can see, or just a minute. Yeah, and in guanidine it is red. So basically here you can see that there is no change, right? And here also this green is like it does not like doesn't show much change, right? So that means again this option 4 is incorrect. So therefore answer here is option 1 and not 4. I hope this answers your this answer of this question is clear to you. Also those who have answered 4, is it clear? Sakina, is it clear to you? And quickly moving over to the last question of this session, which is about microscopy. So let's quickly revise about the microscopy techniques also. Not very difficult questions you will expect from microscopy. Either the questions will be on the terminology like magnification, refractive index, resolution, right? Or you will get the questions where in the options different microscopes will be given to you, and the question will be about a reporter, identification of a reporter or co-localization, right? So which microscope you are going to use. So one such question is on your screen, that in order to visualize intracellular organization of a cell, one can utilize various microscopy based techniques. This include differential interference, phase contrast, dark field, scanning, epifluorescence, transmission, and confocal. So which of the above mentioned microscopes can be used to study dynamics, intracellular dynamics using live cell imaging? So you have to pick the keywords here. One keyword is intracellular organization, another keyword is live cell. Okay, so for this, everyone has already answered option 2. Very good. So quick. This is an easy question, right? So we have already discussed very uh, difficult questions. So I thought of including one easy one. But even if you are not familiar with the concept, you will not be able to, like sometimes we do blunders in the exam, right? We know the answer, but we choose the incorrect answer. Srivani, you are absolutely right. So see, microscopy, when you talk about, we have different type of microscopes, right? Bright field, dark field, phase contrast. So quickly, if I tell you about the keywords, always remember if in any question they'll ask you that in which of the following microscope you can use to get a better contrast. So always the answer will be phase contrast, because phase contrast microscope is used to get a better contrast of your image, right? So basically it converts the like shifting the phase convert into brightness, and you can view the cells without staining here. Fluorescent microscopes always make use of fluorescent dyes, and here you can, like if you want to detect a particular protein in the cell or any protein like with the antigen, so with the help of antibody labeled with fluorescent dye you can detect, right? Immunofluorescence we call it as. Confocal, they help to eliminate out of focus light and increase the optical resolution. And even it helps in co-localization. That is, if two, let's say, proteins are very nearly located in the cell, you can easily differentiate them by using confocal microscope. Then even differential interference contrast microscope, it is used to get a better contrast. And you can use it to generate 3D image, right? And then you have electron microscope. Now the basic difference between light and electron is that live cells can be viewed using light microscope, but electron microscope can never be used to view live cell. Why? Because electrons have very high energy, right? They are of 100,000 times shorter wavelength than the photons of light. They have very high energy and results into killing of the cell. So in electron microscope we use electron beam. right? In light microscope, the source of illumination is light. In electron microscope, it is the electron beam, right? So as in the question, it is mentioned that which of the microscope can be used to study intracellular dynamic using live cell. So always remember, be it any electron microscope, you cannot use it to visualize the live cell. So that means any option other than E and F will be the answer. So in option 1, EF is given, 3 also and 4 also EF is given. So they cannot be the answer. So answer here is option. So this is all about the questions, right? So basically make sure that you revise all the concepts, and whatever concepts I told you today, you just revise them. Okay, don't start anything new at this moment. Okay, so I hope you find the class helpful. Was it helpful? Did you like the session? Yes, was it helpful, students? Are you able to revise and are you able to recall the concepts today? Okay, very good. Good to know. Thank you so much, Anusha, Sri Lakshmi. I'm glad you find it really helpful, and others. Niharika, Saumarya, Sri Lakshmi, Anusha, Vanishri. Thank you so much. So, all the very best. And tomorrow again, we'll be coming up with another session where you will be revising another the concepts, and the you will be practicing the questions from other unit. And even we will be having an exclusive general aptitude session for you all. So this is all for now. Thank you, everyone, and all the very best.